guys, welcome to week 3. We are on module 3. I am here again with my all-star student Jack. And so before we get into module 3, which we're going to be going over some even deeper biology concepts, let's talk just real briefly about module 2. We were really learning about, again, the central dogma of biology. DNA goes to RNA, goes to protein. You have heard me say this so many times now, but I am saying this so many times because later in the course we are going to be learning the implications of this scenario that I just discussed. So in terms of the assignments and the lectures and stuff, what was just like one topic that resonated with you?
I definitely liked learning about the DNA structure and replication.
Okay, good.
It's good.
It was personally never one of my favorite subjects, learning the intricate nature of how DNA unwraps itself. But it is important. It is, remember, DNA replication is done in a semi-conservative way. And I'm really excited that you guys are learning this foundation here. So let me look here. We are looking at week 3. We are going into more biology. We can see that session 1 we're going to be talking about the eukaryotic animal cell. Now, eukaryotes are what we are. Eukaryotes are plants, eukaryotes are animals, okay? And we have another branch of cells called prokaryotes, which are bacteria. So in terms of studying the mind and studying neuroscience, we are going to be focusing on us. It is immensely important, the roles that bacteria play in our world and stuff like that, but we need to focus on eukaryotes. So for the session 2, we're going to be looking at membranes. Now, membranes are the outer entities of cells. They are made of what's called phospholipids. And in many ways, I have a close friend at UC Berkeley. His name is Professor David Presti, and he often said to me, if I was going to have 2 diagrams on my wall, I would have a diagram of the phospholipid membrane, and I would have a diagram of the periodic table, because these things are the 2 most beautiful structures that I know. And one reason why the phospholipid membrane is so beautiful is because it acts, Jack, in a dynamic nature. They are constantly moving around, and they form to the cell's shape. Now, they also allow the cell to retain water in different ways, which we're going to be learning about. We're going to be learning about how water and different molecules are transported across those membranes. Now, how transport occurs across these membranes, okay, is something that is very, very important to neuroscience, which we're going to be covering soon, which we're going to have sort of a longer session on, okay? Now, interesting, I just thought I would just say a few things more about phospholipids. If we get, phospholipids and we just put them in water, what they will do is that they will self-assemble into a membrane. And that's because there's a hydrophobic portion of the phospholipid and there is a hydrophilic part of the, okay. And they line up in this beautiful way to form what are called micelles. And there are these enclosed structures that we can actually see under a microscope. And you'll be learning all about that. Okay, so we're going to be moving on to session 3. We're going to be looking at energy, because there is no biological process, Jack, in our systems that don't require energy. So when we eat food, we are taking in things like glucose, which is like sugar. We are taking in carbohydrates, again, sugar, fats, proteins. And these things are broken down for energy, and they turn into this beautiful molecule called adenosine triphosphate, or ATP. And ATP, I will just say briefly, is a beautiful molecule with 3 phosphates on this molecule. And a phosphate is, again, an element on the periodic table, and it has some oxygen atoms around it. And what happens is, you don't have to know the the nitty-gritty stuff about this, but I just want you to realize that once a phosphate group breaks off of that ATP, there's a whole lot of energy release. Have you ever seen those firecracker things that they're like for the Chinese celebration where they're on a string and there's like a little bit of gunpowder in the middle,
Yeah.
and you pull the string and it's like pop.
We light those off a lot.
Right, you light those off.
Yeah.
So let's use that again to describe ATP. As we're knocking off one of these phosphates, we're breaking this bond and a bunch of energy is released. And that energy is used to drive cellular processes. Right now we are using a ton of ATP. Your heart is beating, you are breathing. There's so many things that you don't even think about in your biology that are going on. So I am so excited for you to learn about how energy is metabolized and how, because again, this is how our brains work. Our brains use a tremendous amount of energy, and this is all related back to what we're going to be talking about later in the course.
Now, can ATP be created in other ways than consuming food?
There are processes in our body where ATP can be synthesized without consuming food, but we're generally consuming some other part of our bodies. You know what I mean?
Yeah, exactly.
So if we're starving, then we're breaking down muscle tissue, we're breaking down fats. So no, the answer is no. You need to be putting in different energy sources, different biological materials. Something that occurred to me that's just interesting that you don't even think about, Jack, is that you're always eating some food that you eat was always once alive. I mean, that might seem obvious to you, once you think about it, but I was thinking about it a few years ago when I was taking my introductory biology courses, and I was like, whoa, that's crazy. You can't eat a rock, literally. There's a ton of energy in a rock. I'm serious. Like, there's so much energy in those bonds, but we just do not have the biological machinery to consume a rock. So don't try it, okay?
Usually don't.
Okay, so that was session 3. Session 3 is energy. And session 4, we're going to get a taste of anatomy and physiology. Anatomy is this dreaded course that a lot of pre-medical students take, where they have to memorize like every bone in the human body. I believe there's 206. They have to memorize different muscle tissues. They have to memorize different organ systems, and it's kind of a regurgitative type of class. We're not going to be doing that here. We're going to be getting down the basics, and we're also going to be studying physiology, which is really more of the study of the relationships between these organ systems and how they're working with each other. So again, that's going to be Hank Green who's going to be explaining that. And in our self-development and reading, what we're going to be looking at is an awesome guy named Craig Venter. Okay, and Craig, I'll just preface this by saying that while we're learning the basics here, number one, we're learning them in a fun way with Hank Green, because Hank is awesome, and Khan Academy, and number two, we're going to already start applying what we're learning right away. Okay, so Craig Venter is this incredible scientist. And what he did is that he was actually able to synthesize DNA from scratch, okay? And he created what's called synthetic life. Now, this has a lot of implications. You guys are gonna be able to understand what he did through, you're gonna watch a 60 minutes segment on him, and you're also gonna be watching a small TED Talk, a press release TED Talk that he gave on this issue, but this has a lot of implications for society. So let's talk a little bit about what synthetic life is. Okay, so what Craig Venter did is that he was able to synthesize DNA from scratch. So you guys learned in the previous module that DNA is made of base pairs. It's made of these different molecules that make up DNA, and there's 4 of them. There's A, C, T, and G. And depending on the order that those base pairs are in, that makes you. We've talked about, again, the central dogma of biology, that the way our DNA is structured is going to literally translate into who we are. It's going to make the proteins that make us. Now, let's talk about this concept of GMO, or genetically modified organisms. Let's not get into the controversy of GMOs right now, okay? But let's talk a little bit about the process of what that means. So what I'm going to explain to you in a part of my neuroscience lecture coming up is that that we can actually get a gene from a different organism and insert it into our organism that we desire to change. So say that we want a tomato that lasts longer on the vine. Okay, we're going to get some sort of gene from another plant that was already there, and we are going to insert it into that tomato. And now that tomato is expressing the proteins that are going to make it last longer. Okay? So in a lot of times in molecular biology, we'll go through this again because this is an important concept. We need to make things light up and we need to make things visible. There's this awesome jellyfish that glows down in the ocean, and what happens is we can take that gene and we can make cells in a petri dish express that gene, and it's called green fluorescent protein, and we can make those cells fluoresce because we took a gene from another organism. Okay, back to Craig Venter. Let's take what he did and see how that's different from just a traditional GMO.
Okay.
He created his own genes. He did not take the genes from another organism. He synthesized his own DNA from scratch. He then got a host bacteria, okay, and he was able to insert his genetic material, his synthetic genetic material, into another organism, and he watched as that organism changed before his eyes. So this is not possible because you are too complex, sir. But say I was able to find a, you told me before, you said, man, I have a really terrible short-term memory. Well, luckily for you, as we'll learn, short-term memory is, probably we know that short-term memory is due to environmental factors that you can change because your brain is plastic, number one. But number two, it has to do with your genes. We're all a little bit different. What if all of a sudden I could insert the genes into you that would help your short-term memory be better? Okay? That is crazy. That is the crazy thoughts, okay? But this is what Craig Venter did on a bacterial level. So again, we're learning the basics here, but we're diving in immediately, applying what we just learned to real life scenarios. This happened, I think, only in 2011. Okay, this was a big deal when it happened. So I'm excited that we've got to this point now that we're applying what we're learning. And this course is going to become more like that as we move on. We're going to be applying the science to real life scenarios. So hang in there for week 4, 5, and 6, where we're going to be talking about the brain. And I'm so excited that you guys are on this journey with us.